Hi there. In this last video on DNA sequencing, we'll talk about some of the new DNA sequencing methods that have increased DNA sequencing throughput so much and reduced costs so greatly. We'll specifically talk about eliminating the expensive steps associated with traditional Sanger sequencing. We'll talk about some of the new amplification strategies and some of the new sequencing strategies. So here are the steps associated with sequencing a genome that we discussed in video 2. I've highlighted several of these as being expensive steps that if we would like to reduce costs, we would need to eliminate. And many of these involved cloning the fragmented DNA into vectors, keeping track of a library, preparing DNA of all those. These 4 steps have been eliminated. The 5th step, the sequencing reaction, the costs have been reduced by miniaturization and multiplexing. You still have to pay for enzymes and nucleotides, but if you use smaller amounts, that's one way of saving cost. And if you can perform millions of reactions simultaneously rather than 1 tube at a time, that also leads to tremendous savings. While the newer methods of sequencing may have started in the late 90s, the real bloom of alternative technology started around 2005. And fortunately for all of us, the innovation continues. We'll talk about a couple different methods, mainly cyclic array sequencing, electrophoretic methods, real-time observation of single molecules, and we won't really get into the details of a couple other methods, but people are working on sequencing by hybridization and ligation. So in this page, we're going to compare traditional Sanger sequencing in this column with one of the new technologies, which is frequently referred to as cyclic array sequencing. But let's review the traditional approach. We have a genomic piece of DNA. It gets fragmented. It used to be cloned into plasmid, put into bacteria. From each bacteria, plasmid DNA would be isolated and purified. You'd do the sequencing reaction, and then you would read out the base pairs. So we want to eliminate the expensive cloning steps and the handling of the DNA and the miniaturization of the sequencing reaction. So here we're going to fragment the DNA just as before, but rather than putting these into a plasmid, what we're going to do is adapt on primer sequences by ligation. So now that all fragments of DNA, not just the blue ones, the yellow ones, the teal ones, the gray ones, have the same primer on them, and then with a simple PCR reaction we can amplify all of those fragments of the library. If we can take these individual molecules and scatter them across a surface and then amplify them, that we can create an unordered array in which each of these spots here on this graph represent millions of copies of a single original molecule. Then what we'll do is we'll add different colors of bases one at a time. We'll record the images and we will build up, say for a specific spot, and you can't pick one out here, but let's say that one, and we'll over time record what the colors are and what the bases are and build up a sequence that way. We've miniaturized the sequencing reaction by having millions of different sequencing reactions on a single small volume, and they're separated by spatial location, so that even though the reagents are mixing for all these spots, the fluorescence that's incorporated at a particular location can be read independently. Now I'd like to focus in first on different mechanisms of DNA amplification, which again is going to be based on ligation of adaptor sequences and then PCR amplification of the double-stranded DNA. Let's first talk about the example that we've already said a little thing about, and that is the spatial location of molecules on a surface. This surface is prepared with lots of PCR primer type molecules, both the blue ones and the red ones. Then we're going to take our template DNA, which we've fragmented and ligated on adapter sequences, and it's going to hybridize somewhere on the surface. We are going to dilute the surface such that the two molecules will on average tend not to be too close to each other, but rather will be separated. Then we'll do a PCR reaction, but we'll do it under specific conditions where we're not adding in lots of additional excess primer. We're going to use the primers that are on the surface, so that the only way for a molecule which binds to the surface to get amplified is for it to bend over and hybridize with one of the other alternative primers that are present on the surface, and then it can amplify. And although that might seem surprising initially, this will happen locally many times for a particular single molecule. We've had a single molecule that's been folded over many times, binding to primers on the surface, which are obviously at higher density than pictured here, and these molecules will be amplified. They'll be geographically close to each other, and they won't interfere with a second molecule which bound over slightly to the side on the surface, which is also going to be amplified. And so what we have is what's sometimes called a colony of PCR products. Colony because that's what appear on a plate, but these are PCR products, so they're sometimes referred as polonies. Now we've amplified a single molecule into perhaps millions of copies of that same one in a location, and we're going to eventually sequence this using fluorescence imaging. Another type of PCR can happen on a microsphere bead, which has a single primer attached to it. The second primer can be in solution, but what you're doing is you're using a method called emulsion PCR. We'll have aqueous droplets inside this hydrophobic solvent, and each of one of these aqueous drops is going to act like an independent test tube. You're not going to have mixing of one of the components of one of these aqueous drops with one of these other drops, because the molecules are not thermodynamically stable in the hydrophobic solvent. Then we're going to perform PCR with one of the primers attached, and in the end you're going to have a microsphere with many copies of a single piece of DNA attached to the sphere. And then these spheres can be distributed on a glass slide, perhaps chemically cross-linked. I don't think it needs to be. But in both of these cases, in approximately a centimeter square, you might have as many as 200 million different clusters, allowing you to perform 200 million sequencing reactions in a very small volume. Now, there are two sort of competing single molecule approaches where amplification is not needed. Here, these original molecules that had these adapters ligated will bind to a surface. We'll sequence these individual molecules, and we'll just use extremely sensitive optics and detection to recognize fluorescent signals for each of these individual molecules. The final approach that we'll talk about is not attaching the DNA to a surface, but attaching the DNA polymerase to a surface, and then having DNA molecules thread through the DNA polymerase, but be spatially localized because of their interaction with a polymerase. So when we look at the sequencing reactions, we'll start by talking about this example where we had single molecules amplified on the surface, creating polonies. So just as a reminder, this is the amplification that we're talking about. Now here we see three different polonies, and they've only shown one DNA molecule for each of these, but we have them attached to the surface because they were amplified with primers attached to the surface. We're going to put a sequencing primer on there. We're going to start doing base pair extensions, so we'll do the sequencing reaction right there on the surface. We'll diffuse in 4 different fluorescently labeled terminator nucleotides, but they have a twist. They're terminators, but they will eventually be reversible. So the first thing that's going to happen is you'll incorporate the different bases at the different locations. Because they are terminator nucleotides, you will not be able to add a second. You'll take an image, a photo, of which locations incorporated which color, and then you're going to treat these chemically. It's going to cleave the fluorescent molecule. It's going to cleave the protection that prevents it from being a terminator, and now you are reconstituting an active 3' hydroxyl group. Now we're going to add a second batch of these fluorescently labeled reversible terminators. We'll add a second base. We'll take a second picture, and then we will again treat chemically to remove the fluorescence and unblock the 3' hydroxyl, allowing for further incorporation. Now if we look at these two sequences, there's two that are circled here. There's one on the top, there's one on the bottom, and here's the color key. So we can see green, blue, red, green. Using the legend, we can see green, blue, red, green is CATC. CATC are the first 4 bases, and then we have cycle 5 and cycle 6. And at the same time, we recognize this particular polyny down here at the bottom. And we see green, 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 green. And this is a homopolymer repeat of C, 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 C. And you can do that for any of the fluorescent spots that we observe here. And again, with 200 million of these spots in a 1 centimeter square, generating a great deal of sequence information for every single base that's added. Now these methods are not capable of producing thousands of base pairs in a row. These reads are very short. They can be as short as 32 base pairs. But if you do 32 base pairs for 100 million spots, that's going to be 3.2 gigabase pairs, which is approximately the size of a human genome. So you can see how that scales very effectively. Another sequencing method is being promoted by Pacific Biosciences called, their acronym is called SMART sequencing, for single molecule real-time sequencing. Again, we have a sequencing by synthesis type approach, but here we've isolated polymerases, attached them to the surface. And the surface is in the presence of wells, and so you have a pretty significant depth here on a molecular scale. And what we're going to use is epifluorescence, meaning we're going to excite with an evanescent wave, and the light is only going to extend a certain distance into the well here. So in this case, only nucleotides that are in a certain proximity to the surface will get excited using the light, and only those nucleotides that are being incorporated by the polymerase spend sufficient amount of time in the well to produce a lengthy signal, and this sustained fluorescence for a long period of time is what gets called as a base. On these dimensions, if a different molecule were to diffuse in, it would diffuse out rapidly enough so that it wouldn't be called as a base pair. We conclude with a discussion of one of the potentially most exciting methods, and that is called nanopore or electrophoretic sequencing. And here we have a lipid bilayer, we have a channel molecule which actually imports DNA into the cell, single-stranded DNA, because the 4 bases are different sizes, that as an individual base goes through this narrow pinch point region, it's going to restrict or allow the flow of different amount of accompanying ions like sodium or chloride or any of those. And there's going to hopefully be a characteristic signature in terms of how many different ions can pass through when a different base is passing through here. One of the very exciting aspects of this is that in principle you might be able to get many thousand base pair reads, because DNA is known to be transported through these channels at those sizes, so that would make sequence assembly much simpler. And again, this is hypothetical readout, we'll stress, because they've been working on this for so long, but you might be able to read different bases based on the amount of ion current that can flow at any particular time when a particular base is passing through. Whether this method would ever become competitive with some of the other methods that we've shown remains to be seen, but it would be wonderfully complementary if it would, with the long reads and the comparatively very fast sequencing times. Okay, thank you very much for listening.